afternoon, everybody. I'm really glad to be here today to present you my PhD works on the behavioral responses of cetaceans to natural and anthropogenic sounds, which I did under the supervision of Valerie, Patrick, and Charlotte. For the next 45 minutes, I take you into the marine environment, where sounds travel 5 times faster than in the air, and they suffer little absorption in seawater, especially at low frequencies, and therefore the soundscape of the ocean is very composite, and the sounds from a multitude of sources intermingle. And in this environment, cetaceans, they rely on sounds for many biological functions. For foraging, with, for instance, odontocete, here a sperm whale producing echolocation clicks to detect their food source. For reproduction, with the example of the well-known humpback whale song on the breeding grounds. And to maintain social cohesion, with a vocal repertoire of group-specific calls to maintain group contact in killer whales, for instance. However, the exact role of these conspecific sounds is not really clear. The same click-based signals that are used for foraging are also used in communication in sperm whales. And where the humpback whale song is involved in reproduction, its exact role remains a mystery, with the songs of humpback whales attracting males and repelling females. And for some species that have really stereotyped sounds, it's easy to describe their vocal behavior, or relatively easy to do so, and to detect group-specific signatures. But some species, such as pirate whales, have graded repertoires that vary along a continuum, and it's challenging to describe this vocal behavior and find relevant markers in them. All these sounds produced by cetaceans, they travel 5 times faster than in the air and with little absorption, so cetaceans can receive the sounds from other species. And most of the work that has been done on that has been focusing on killer whale sounds, because killer whales are a cosmopolitan species and they are known to prey on most cetacean species. And the other cetacean species show strong anti-predator behavior in response to killer whale sounds, avoiding the source of such sound, stopping their vocal production, or other kind of behavior such as mobbing-like behavior. And some species are able to go a step further in their use of heterospecific sounds, and 2 species have demonstrated an ability to discriminate between different subpopulations of killer whale sounds. The sounds of different subpopulations of killer whales. And another sound source present in the environment that is interesting in this context are anthropogenic sounds, or all the sounds related to human activities, because there has been an increase in human presence and a development in human activities at sea, and that has been raising concerns about the potential impacts on the ecosystem. And there have been studies on these impacts, and several behavioral responses to anthropogenic sounds have been documented. For example, the avoidance of anthropogenic sound sources, the displacement of animals from a biologically important area, and the potential masking of biological signals, be it communication signals or echolocation ones. But these responses to anthropogenic sounds are really variable. They vary between species, with some species being really sensitive, for example, beaked whales. They respond to low received level of sounds with really strong responses compared to other species, such as pirate whale, which display less intense response at higher received levels. So there is a need to better understand the impacts of anthropogenic sounds can have on the ecosystem, and that goes through a better understanding on the vocal behavior of cetaceans and how they are able to use the sounds they can detect from the environment. And for this, we need better tools to describe some of the vocal behaviors for the species with graded vocal repertoires. So the main question in my PhD was how informative can environmental sounds B for cetaceans, with a focus specifically on heterospecific sounds and their ecological implication. And that's the first part of this talk, and that includes my first PhD chapter. And another part on the impacts of anthropogenic sounds on the marine ecosystem. And this regroups the second and third chapter of my PhD. So we can start with the first part. How do cetaceans respond to the sound of heterospecifics? The detection of heterospecific sounds, also referred to as heterospecific eavesdropping, can have fitness enhancing consequences for cetaceans, and the implication of detecting heterospecific sounds depends on the ecological relationship between the species that produce the signal and the species that receive them. For instance, in a foraging context, a prey that could detect the sounds produced by its predator and recognize them would be able to use this information to avoid predation. Whereas a predator that could detect the signals produced by a food competitor could use this information, for instance, to locate the common food patch. And during my PhD, I've been studying the responses of cetaceans to killer whale sounds, because on the one hand, killer whales are a cosmopolitan apex predator, as I said in introduction. So detecting the sounds of those predators can be used to avoid the predation related with them. However, not all the interaction between killer whales and other cetacean species involve predation. Some species are playing, resting, or traveling together with killer whales. Some species feed on the same place and sometimes even on the same food source. And for those species, killer whales can be competitor as well as predator. And this variability in the interaction between killer whales and other cetacean species can be related to the variability in killer whale ecology. Some populations have diverged into different ecotypes that are subsets of this population that share ecological adaptations. And these ecological adaptations can be different diets, with some ecotypes being specialized in marine mammal eating, while some others feed mostly on fish. That means that they are engaged in different ecological relationships with the other cetacean species. And ecotypes can also display different vocal behavior, and that can be a cue, an acoustic cue for the other cetaceans to discriminate between those ecotypes and the ecological relationships they have with them. So it may be adaptive for cetacean species to have this ability to discriminate between the sound of different killer whale ecotypes. This ability has been demonstrated in one species of pinnipeds, the harbor seal, and recently in a species of odontocetes, the longfin pirate whale. And in this first part of my PhD, I investigated whether a species of mysticet, or bilead whale, the humpback whale, was able to discriminate between the sound of two killer whale ecotypes. In order to do so, I did some playbacks of killer whale feeding sounds. The idea is to broadcast the killer whale sounds and to record the behavior of cetaceans before, during, and after the stimulus presentation. So those periods are referred to as the pre-exposure period, the exposure period, and the post-exposure period. And then comparing the behavior of cetaceans between these experimental phases can answer a question. For example, if we compare the behavior of the whales before and during the stimulus presentation, we can identify what are the modification of behaviors elicited by the sound stimulus. And if we compare the behavior before and after the stimulus presentation, we can determine whether or not the whale went back to its behavior. We use the sounds from two killer whale ecotypes. The first one is transient killer whales from the Northeast Pacific. They are marine mammal specialists, feeding on pinnipeds, but also other species of whales. And herring-feeding killer whales from Norway, which, as the name implies, feed mostly on fish. And for humpback whales, the first one can be potential predators, whereas the second one shares the same food source in northern Norway, where we did our experiments, and are therefore food competitors. And those two ecotypes produce different sounds. So here is a sample of mammal-eating killer whale sounds. You can hear the killer whale vocalization. And this behavior is different from what we can observe in herring-feeding killer whales, where the same kinds of vocalization are present, but somewhat higher in frequencies. And there are echolocation clicks in the background that I want you to be aware of. And we broadcasted the sounds of those two killer whale ecotypes in two contexts: of Spitsbergen during summer in offshore waters, and during winter within the fjords around an island called Vengsøy. And for the results, we used this open sea drawing and this fjord drawing to symbolize the experiments. Our hypotheses were that the sound of mom-eating killer whales could signal the risk of predation to humpback whales, and we expected anti-predator behavior to this kind of sounds, so a flight response mostly, and that the sound of fish-eating killer whales could signal the presence of a food patch to the humpback whales. We expected a dinner bell effect here, with the humpback whales getting closer from the source and exploring the water layer around it. And we also investigated the influence of the ecological context, comparing the inshore and the offshore experiment, with the hypothesis that the inshore setting is a closer habitat than the offshore one, and that could restrict the behavioral responses of the whales. To describe the behavior of the humpback whales, we use visual observations at the surface and animal bone tags to record the underwater behavior. So here are the tags we used. They are called mixed tags. They have multiple sensors: a pressure sensor to reconstruct the dive profile of the whale, accelerometers and magnetometers to reconstruct its 3D orientation, and hydrophones to record its acoustic scene. They are non-invasive tags. They are attached temporarily to the animal using 4 suction cups, and we deploy them either with a long pole or with a pneumatic launching system. And using the data recorded by the visual observers and the tags, we describe the behavioral responses of humpback whales in terms of horizontal movement, measuring these horizontal reaction scores that quantifies how much the whale either approached the sound source or avoided it during the playback, by comparing the distance between the source and the whale at the end of the playback with the hypothetical position it would have if it continued using the same movement pattern, general speed and general heading, as it was doing in the pre-exposure period. And we also described the behavioral responses of humpback whales in terms of vertical movement. And for each of the 3 phases of this, pre-exposure, exposure, and post-exposure periods, we measured the maximum dive depth and the maximum dive duration. So here are the results for the horizontal movement. The horizontal score on the y-axis with positive reaction scores, meaning that the whale approached the sound source, and negative ones that it avoided it. There was an approach of the sound source during the fish-eating killer whale playbacks inshore with 6 out of 6 approaches. And overall, the humpback whales avoided the source of mom-eating killer whale sounds, but this avoidance was more marked in the offshore setting with 7 out of 8 avoidances than in the inshore setting, where it was more mitigated with 2 avoidances and 2 approaches. Concerning the vertical movements, the clearest result is that humpback whales dive. So this is the pre-exposure period, exposure period, and post-exposure period. They dove deeper and for longer duration during the playback of fish-eating killer whale sounds, but went back to pre-exposure levels of diving activity right afterwards. And during the mom-eating killer whale experiments, the responses were more variable, with inshore a tendency to increase dive depth and duration during and after the playbacks. Now these results are for fish-eating killer whale sounds inshore, an approach and deeper, longer dives during, which is consistent with our expectation of a dinner bell effect and an approach and explore response. And the horizontal avoidance and variable vertical responses could be consistent with a flight response that would be stronger in the offshore context compared to the inshore one. The two key results here being that humpback whales reacted differently to the sound of two killer whale ecotypes, and that the context of presentation influenced the behavioral response to predatory killer whale sound. There's a few points to discuss about these results. The first one is that we cannot differentiate an effect of the ecotype and the familiarity of the killer whales and humpback whales, because our food competitors were recorded in Norway, where we also did our experiments. Therefore, the population was familiar for the humpback whales, whereas our predator sounds were recorded in Southeast Alaska, and therefore are not familiar to the humpback whale population we tested. But additional playback experiments could help us untangle these effects. Then there were some differences in the inshore and offshore experiments. The response was more mitigated inshore with 2 avoidances and 2 approach. So, but it doesn't appear that the inshore setting was restricting the flight response of the humpback whales, because the 2 avoidances observed in the inshore setting were similar in intensity with what we observed offshore. Another possibility would be that these 2 contexts correspond to different seasons for the humpback whales. During the winter, that's the end of the feeding season, so the whales are in top condition concerning their lipidic storage, whereas during summer, that's the really beginning of the feeding season, so the whales would have more pressure to forage, to replenish their resources, but also they would be more vulnerable to killer whale attacks, so this could have influenced their decision-making process. We observe much variability between the responses of different individuals to the killer whale playbacks. That's a recurring topic in marine mammal science. The fact is that there are many factors that could affect the response, the group composition of the whale, or the behavioral activities that are engaged in at the onset of the playback. And during my PhD, we did not look at these factors in too much details. In conclusion, humpback whales were able to discriminate either between predator and competitor ecotypes or between familiar and unfamiliar killer whales. That's an ability that's also present in pinnipeds and undoctocete, and we can wonder whether this fine-scale discrimination of heterospecific sounds may be widespread in marine mammals. So after this conclusion, we can move on to the second part of my PhD about the responses of cetaceans to anthropogenic sounds and their impact on the ecosystem. Among all anthropogenic sounds, I've been focusing on naval sonars for a number of reasons. They're really intense sounds. They're produced higher than 200 decibels source level. Their frequency range can overlap with signals produced by cetaceans. And there has been, last but not least, spatial and temporal correlation between naval sonar use and mass cetacean strandings. So in order to study the response of cetaceans to naval sonars, we do what's called behavioral response studies. The idea is to do a sound exposure experiment, just as we did with killer whale sounds, but with sonar signals, with controlled condition, in order to protect the whales from the potentially negative impacts of the sonar. So we're using a single sonar source, whereas military exercises could have several of them. We increase the sound levels step by step, and there is an emergency shutdown procedure ready in case the whales show pathological or really negative responses. And all these experiments, during my PhD, I did them as part of a big international research team called the 3S project for synomal sonar safety. The second step in that, after describing the responses to naval sonar, is to assess their biological significance. What are naval sonar perceived as by the cetaceans? And one of the possible approaches to do that is to compare the responses to anthropogenic sounds with the responses to natural ones of known significance. This theoretical framework is called the Disturbance Hypothesis. And the idea is to compare here responses to sonar with responses to a natural signal that corresponds to a risk of predation. For cetaceans, we use killer whale sounds, but just to remind you the result from the first part, killer whales have a very variable ecology, and not all killer whale sounds may be suitable candidates to signal the risk of predation to cetacean species. So during this PhD, I've been focusing on one species in particular, the longfin pilot whale. They are medium-sized odontocetes, 5 or 6 meters long. They live in social group all year long, and their social group are very fluid in organization. The basic unit is 10 individuals, and then groups constantly split and join. And they display high levels of vocal activity, and it is thought that exchanges of vocalization in this species could mediate the fluid social organization itself. So several aspects of the behavior of longfin pilot whales to naval sonar have already been described by the 3S research team, and they've shown that the whales tended to avoid the sound of sonar signals, that they may cease their foraging activity during sonar exposure, and that they tended to form bigger groups. But the vocal behavior of longfin pilot whale has received relatively little attention compared to other aspects, with studies either staying at the global scale of vocal activity or focusing on very detailed behavior, such as the vocal matching of sonar signals. So during my PhD, I decided to focus on the vocal behavior of pilot whale, trying to describe into more details how they responded to naval sonar. And that proved to be a challenging task because first, they live in these cohesive social groups with very fluid social organization, fission-fusion dynamics, which makes it really hard, if not impossible, to identify which individual is producing the calls. And then they produce typical odontocete sounds, so they produce echolocation clicks that are short broadband pulses, a few milliseconds long, that are mostly used for echolocation, but can also have a communicative function. They produce whistles, which are harmonic sounds with really intricate frequency modulation patterns, and pulse calls, which are a repetition of short broadband pulses, so fast that it appears tonal to the human ear. But with longfin pilot whales, the situation is a bit blurrier, because the vocalization they produce can contain portions of tonal sounds, pulse sounds, and broadband sounds, and even some transition sounds that transit from one acoustic nature to the other. Some of these signals are stereotyped and they are produced repeated in the exact same way, but most of them are graded and vary along a continuum, which makes characterizations challenging. And they have a very large and diverse repertoire. The latest attempt to describe their vocal behavior, focusing on the stereotype sounds, define more than 120 call types. So I decided to develop a new method to describe this. The usual method is to have trained operator classify manually the sounds by listening to them and by visually inspecting the spectrographs. This comes with an amount of subjectivity because human operators decide what is the boundary between call types, and it is a really time-consuming process. To give you an idea, the data set I worked with during this part of my PhD contained 14 tags deployment, 37 sound exposure experiments, and that amounts to more than 40,000 calls. So it wasn't realistic to manually classify this amount of data. Instead, I implemented an unsupervised classification algorithm. My two main objectives were to have an objective and reproducible classification procedure, and that was faster than the manual classification. I did so by using male frequency capsule coefficient to describe the calls, and then a fuzzy clustering algorithm for the classification task. Male frequency capsule coefficient are a mathematical tool that comes from human linguistics, that has been increasingly used to describe animal vocalization. The two key points to remember about this method is that it is an effective compression method. It summarizes much information about the distribution of energy along the frequency spectrum in a small number of coefficients. And then it is defined for all kind of acoustic natures. It's defined for harmonic, pulse, and broadband sounds, which makes it really suitable for long-fin pilot waves, because the calls they produce can have portions of different acoustic nature. For comparison, some studies rely on fundamental frequency-based measurements, but those only exist for sounds that have fundamental frequencies, not for broadband sounds, for example. And then the fuzzy clustering algorithm has 2 points going for it. It's a soft classification algorithm and it's unsupervised. So first, what is a soft classification approach? Most usual classification approaches are hard classification. They determine absolute boundaries between call types, and one call is assigned to a single call type. So for example, you decide on boundaries between call A, B, and C, and it doesn't matter how close a call is from the boundary or how far it is. On one side of this line, your call type A, and on the other one, your call type B. Instead of doing that, the fuzzy clustering detects from the distribution of the dataset apparent stereotypes that becomes the center of the different categories. And then each call is given a membership score, a likeliness to belong to a cluster based on the distance between the call and the different clusters. For instance, all the calls here that are much closer from centroid B than from centroid A and C may be 90% B and 5% A, 5% C. Whereas the calls that lie in between centroid A and C and much further away from centroid B may be 40% A, 40% C, and only 20% B. And the key point is here that one call is given one score for each of the clusters in the dataset. I'm going to show you the results of the fuzzy clustering for illustration. So the first thing is that the results from the fuzzy clustering, there isn't a single solution with clearly defined clusters. Instead, depending on the fuzziness parameter, which dictates how much clusters can overlap, if you have a really high fuzziness, then the cluster overlaps so much that they're actually the same one with all the calls in them. And if your fuzziness is really small, close to 1, that's equivalent to hard clustering. And in between, some solutions are going to be stable over some interval of fuzziness. And we consider this stability to come from the fact that these solutions are describing an underlying structure in the dataset. So here, for instance, we have a 2-cluster solution, a 3-cluster solution, and a 4-cluster solution. They're all valid. They just describe the dataset in a different way. And if we take, for example, this 4-cluster solution, we can use a PCA to visualize the spread of the dataset in 2 dimensions. You've got the 4 plus crosses that are the center of the clusters and the distribution of the data point with it. We can use the coordinates of the cluster centroids to reconstruct the stereotypical call. For example, here is the center of the blue cluster, and we can use them to say that a typical blue call would look like that. We can do that for every cluster. And you can see, for instance, that here, the highest overlap we observe is between the blue and the green cluster, and they are much closer in structure than with the red and the orange cluster. And we can also quantify whether the calls are close from a cluster and are stereotype calls, or whether they are graded ones, measuring the typicality, which is a difference between the 2 highest membership scores. High values of typicality mean that the calls are much closer from one cluster than from the other ones, and therefore they are stereotyped calls, whereas low value of fuzziness of typicality means that the call lie in between clusters. For instance, you can see that the red cluster tend to have high values of typicality, and if you look at the red cluster, those points are closer from the red cluster center than for the other centers. Whereas if you take the blue cluster, you can see that it has much lower values of typicality. And if you look at the spread of the data, you can see that the blue cluster is in between the green, orange, and red one. And those calls are approximately as close from the blue centroid than from another one. In conclusion, the method was validated. We tested it with real long-fin pilot wave recordings, and we obtained stable clustering solution. Compared to other methods, we have a lower precision in terms of number of categories than what the manual classification can achieve, and a similar thing to other unsupervised art classification algorithms. Another difficulty is that there are several clustering solutions that describe the data set in different ways, so it makes a bit more complex to analyze these results. But in conclusion, both our objectives were achieved. Our classification is much faster than the manual classification procedure. We took a few days to run the fuzzy clustering algorithm, whereas we could have spent months to manually classify the data set. And it takes into account the graded nature of long-fin pilot wave vocalization and in a quantitative way. So now that we have validated the method, we can use it to describe the vocal responses of long-fin pilot wave to neural sonar, and then compare these responses to the vocal responses to killer whale sounds to try and evaluate how neural sonar may be perceived by long-fin pilot waves. We did so by using a sound exposure experiment with neural sonar signals. The idea is the same one as with the killer whale sounds, with the pre-exposure, exposure, and post-exposure periods. And we used 3 kinds of sonar signals that vary in frequency band and frequency modulation pattern. The first one is a low frequency sonar rising from 1 to 2 kilohertz. The second one is a medium frequency sonar rising from 6 to 7 kilohertz, and that may hurt a little. Sorry about that. And the last one is a low frequency sonar from 2 to 1 kilohertz, and this one should be okay. So when we present the results, I will use these simplified spectrograms to identify which kind of sonar signals we're talking about. And we compared these responses to sonar signals with responses to killer whale sounds. We only use transient killer whale sounds since they are marine mammal specialists and should therefore be suitable to signal the risk of predation to long-fin pilot whales. Our main hypothesis was that long-fin pilot whales would display coordinated response to threatening stimuli, neural sonar and killer whale sounds, and that vocal exchanges of vocalization would mediate these coordinated responses. Therefore, 2 implications in terms of vocal behavior. We expected an increase in vocal activity during the exposure, and then if there were some stereotyped response, such as what we can observe in birds or terrestrial mammals, stereotyped alarm calls or stereotyped recruitment calls, we would expect a change in the types of call produced during the exposure to neural sonar and killer whale sounds, and an increase in the call stereotypy. Now, was there an increase in vocal activity during the exposure? Yes. In response to the low frequency 1 to 2 kilohertz sonar, we observed an increase of vocal activity during and after the exposure. For the other types of sonar signals and killer whale sounds, we observed variable changes. There were changes, but the direction varied across experiments and between type deployments. Anyway, these responses were different from what we could observe during control experiments. We did sonar exposure experiments without actually broadcasting any sonar, and those are called no sonar controls. And we observed a tendency to decrease vocal activity after these experiments. And we did some broadcasts of broadband noise as a control for killer whale playbacks, and we observed the same tendency to decrease vocal activity during. So in conclusion, we observed an increase in vocal activity to one type of sonar and variable changes to the other acoustic stimuli we tested, and these responses were different from what we observed during the control experiments. Moving on to the changes in code type usage. The whales did change the types of code they were producing. So here you have the results from the fuzzy clustering with the 3 categories obtained for this particular individual. And here you have a stacked histogram of the call membership scores along time with the pre, during, and post periods. And what we can see is that there is a clear switch from mostly green calls here in the middle to mostly blue calls during the exposure to 1 to 2 kilohertz sonar. We observe similar changes to the medium frequency sonar from 6 to 7, with here 4 clusters for this individual, and a switch from the orange one to the red one. We also observe changes in response to the 2 to 1 kilohertz sonar, and some changes in response to kilowave playbacks, with here an increase in the membership scores to the blue cluster during the playback. So in conclusion, there was a change in code type usage in response to all the acoustic stimuli we tested, as well as all the control experiments, but there was no consistency across experiments. We could not detect stereotype responses in response to the sonar or in response to kilowave sounds. We also looked at the code typicality, the idea being that if there were stereotype alarm calls or stereotype recruitment calls, those would be repeated in a highly conservative way and would therefore have higher typicality than the other calls of the long-fin pilot whales, which are more graded. However, we did observe changes in typicality, but nothing directional. So, there was an increase in vocal activity in response to one type of sonar, and variable changes to the other stimuli. Changes in code type usage to all experiments, but nothing stereotyped, very variable changes, and variable changes in typicality. So the main result here is that we observed variable changes in the vocal behavior of long-fin pilot whale in response to acoustic stimuli, and we could not identify stereotype responses. Therefore, there were changes in vocal behavior, and since we have observed other responses, behavioral responses of long-fin pilot whale to sonar, and since the vocal behavior of pilot whale occupies such a central place in their ecology, we can assume that naval sonar has an impact on the behavior of long-fin pilot whale. However, we're still lacking knowledge about the baseline behavior of long-fin pilot whale. What are the different code types they produce? What are their functions? What are the functions of the code gradation? And we still need more information about this baseline vocal behavior to fully understand what is the impact of naval sonar on long-fin pilot whales. Moving on to the general conclusion. The main question was, how informative can environmental sound be for cetaceans? We found evidence that there were fine-scale discrimination ability of heterospecific sounds in a mysticet species, the humpback whale. And this ability is also present in pinnipeds and in odontocete. So we can ask ourselves whether marine mammals in general have this kind of fine-scale acoustic discrimination abilities. The ecological context of the sound presentation influenced the response to heterospecific sounds. And there are many factors in this ecological context that could be of interest and would require further investigation. And we were able to detect impacts of the exposure to anthropogenic sounds on the vocal behavior of a species with an especially challenging vocal behavior to describe. But the response was not stereotyped, and we're still lacking the basic understanding of their vocal behavior to draw a conclusion about our observations. in perspective, we still need more sound exposure experiments. We could use them to entangle the effect of the ecotype and the familiarity of the killer whale sounds we used in the first part. And we could also use it to explore the influence of the context of presentation on the responses to anthropogenic sounds, for instance. The phasic clustering method worked, but there is still room for improvement. The calls of long-fin pilot whale can be composed of several subunits or portions of different acoustic nature, and it should be possible to include this temporal resolution within the method. And we're still lacking a whole lot of data about the baseline vocal behavior of pilot whale to fully understand how it functions. So I hope that during this talk, you picture that the use of sounds by cetaceans were not restricted within a species with communication signal from one individual to the other one, but was much more composite and complex, with cetaceans being able to detect and to process sounds from heterospecifics, having responses that were adapted to the ecological reaction between species, and that the anthropogenic sounds were also having an impact on the behavior of cetaceans. I would like to thank my supervisors and all the research team I work with, my host labs and the 3S team, the PhD committee and the referees for their feedback on my work, all the co-authors I had the pleasure to work with, and my friends and family, which made it possible to finish the PhD with a bit of sanity left. Finally, thank you for your attention. If you have any questions and a PhD, I will be happy to answer them.